The broadcast is now starting. All attendees are in listen-only mode.
Good afternoon and welcome to our webinar for today. My name is Glenda Cleasy, Agronomy Specialist with Saskatchewan Pulse Growers. And with me today is Andrea Lauder, Communications Manager with Saskatchewan Pulse Growers. And thanks to Andrea for all of the work in organizing the webinar for today. So for those of you on the line, welcome to the fourth and final webinar for this 2018 growing season as part of our Pulse webinar series. As a reminder, you do get CCA or CCSC credits for attending this webinar live. To get CCA credits for this webinar, again, you must be watching it live. Andrea will send out an email after the webinar requesting your CCA number. At this time, please respond with either your CCA or CCSC number. If more than one person is watching from one computer, please make sure you verify all of those in attendance, and then you can include all of their CCA or CCSC numbers. Webinars will be recorded, and they will be posted to Saskatchewan Pulse Growers' YouTube account. For those of you who have been on our webinars before, this is a new update and a new change, so you can now find all of the webinars on the Pulse Growers' YouTube account. This will allow you to view them in the future, or if you know someone who was unable to attend, they can go there to watch it, or you can look back on the material that was covered. For today's webinar, all participants will be muted. We will be happy to take any questions. To ask a question, please type it into the question box located in the GoToWebinar dashboard. You can send questions at any time. If it's important to ask the question at the moment, we will do our best to fit it in as we can. Where a question can wait, we will hold that question until the end of the presentation. Our presenter today is Dr. James Tansey. Dr. James Tansey has been the Saskatchewan Ministry of Agriculture's Provincial Insect and Pest Management Specialist since January of 2018. Jim grew up in St. Albert, Alberta, and graduated from the University of Alberta with a bachelor's degree in biology, a master's degree in ecology and environmental biology, and a PhD in entomology. He also completed 2 postdoctoral fellowships. The first was at the University of Alberta, working on insect-plant interactions and host plant resistance in canola. The second was with the University of Florida, working on citrus entomology. His research background includes integrated management of insects in field crops and tree crops, insecticide resistance and efficacy, the biological control of weeds and insects, and attenuating populations of urban pests like house dust mite and cockroaches. He has also taught biology and worked in regulatory affairs. James is going to speak to us today on insects and scouting for insects in pulse crops. So thank you, James. I will now turn the presentation over to you.
Great, thanks. Thanks to everybody for attending, and I'll get on with the show. So just a brief outline of what I'm going to talk about today. We'll talk about some important pests of pulse crops in Saskatchewan. This isn't an omnibus list. This isn't every pest that you're going to see in pulse crops, but it hits some of the high notes. We'll talk about recommendations for control, in addition to some of the identification and biology of the animals. We'll talk about some beneficial insects, that is, natural enemies of some of these pest insects. We'll talk a little bit about the preliminary pea leaf weevil survey results from Saskatchewan, and we'll wrap things up by talking about the regulatory status of some insecticidal seed treatments important to some pulse growers. So we'll get rolling. Pulse pests, the first we'll talk about is the pea leaf weevil, Cytona lineatus. Adults are the overwintering stage. They're active right now, so they're emerging in the spring. Their characteristic notch feeding can be seen on alfalfa, clover, dry beans, faba beans, field peas, pretty much any legume that you can imagine. And if you look at the lower right photo, you can see that characteristic notch feeding on a pea seedling. The photo directly above, of course, is the adult, and the photo above that is a larva on a root nodule. This notch feeding can be damaging to plants, although highly unusual. It can kill plants. Highly unusual to kill large numbers of plants, that is. Can kill plants, can reduce pod production, and females are highly fecund. So females are dropping eggs as they walk along the soil surface in small clumps, typically 1 to 24, and they'll continue laying eggs and over the course of the season and can produce 1,500 eggs per female, so highly fecund. Larvae are the more damaging stage, and reason being is they consume the rhizobium in the nodules. They have some host limitations and do best as far as completing development on field peas, although the adults will feed on other legumes as a food source. Of course, consuming the rhizobium is going to have an effect on nitrogen fixation, and heavy populations, very heavy populations, can lead to 27% yield losses. So once again, top right is the larva on a nodule, below that the adult, and some notch feeding. Scouting for pea leaf weevil should be done at the 3 to 6 node stage, so right about now, late May to mid-June. You're looking for 10 locations per field, each of these 25 meters apart. So if you look at the figure, you can see that there's a line of sampling sites 100 meters into the field, another line of sampling sites 2 meters into the field, so 5 per. You want 25 meters in between. Look at 10 seedlings per location. And what you're looking for is that characteristic notch feeding, both on the leaves and the clam leaf in particular. Yield losses are associated with 30% of seedlings having damage to clam leaf. The tricky question is whether or not this is actually an action threshold, and I'll talk about this in the next little bit, or in the next slide, pardon me. So some means to control pea leaf weevil include cultural control, so crop rotation, reducing the amount of plants for population increase. There's recent evidence that nitrogen fertilization can help, which I realize sounds a little counterintuitive with a pulse crop, but it can help with yields, as can seed inoculation. Reduced till or zero till can also reduce adult movement, and think of things in terms of, given an option between running across a football field or running across, a fallen tree stand. There's going to be physical limitations to their movement. A lot of their invasion of crops is done by walking. So you're going to get physical limitation to their movements, plus it presents a lot of places for natural enemies to hide. So running through a jungle full of tigers. There's also early planted border strips can be an effective means to concentrate populations. So they'll come up a little bit earlier, present a food source to the adults, concentrate those populations, and let you knock them down in a relatively small area. So chemical control, there are a number of products listed in the 2018 Guide to Crop Protection. For those of you who don't refer to it, I encourage you to. I mentioned earlier some potential issues with action thresholds associated with this weevil. Mixed results from applications of foliar insecticides from a yield perspective. You can kill the adults, but foliar application isn't going to have an effect on the eggs and isn't going to have an effect on the larvae. And of course, the larvae are the damaging stage. So once again, mixed results with foliar insecticides. The best results are with seed treatments, particularly with the neonicotinoid thiamethoxam. The need to, of course, to apply seed treatments needs to be weighed when you're making your seasonal planting and purchasing decisions. And it's important to base treatment with seed dressings on the previous year's population. If you're in a hot spot for pea leaf weevil, neighbors have suffered a lot of damage, you may want to consider using a seed dressing. Some natural enemies of pea leaf weevil, both their eggs and adults are susceptible to predation. Small ground beetles, including Bembidium quadrimaculatum, that's the top right picture. That's a tiny little generalist predator who loves insect eggs, including the eggs of pea leaf weevil, and they'll munch them right up. Large ground beetles and wolf spiders are also generalist predators, and they will eat whatever they can tackle, and that includes pea leaf weevil adults. And so the ground beetle pictured lower right is Pteraspis, and a wolf spider pictured lowered left. There are also a number of other generalist predators that will attack adults. The next pulse pest we'll talk about is Pea Aphid. And unlike the pea leaf weevil that actually damages tissues as an adult to consume them, these ones insert mouth parts into the leaf surface and draw plant fluids. Typically, which is called piercing sucking feeding. They have piercing sucking mouth parts, somewhat analogous to the way mosquito feeds. They typically feed on the lower surface of leaves and pods, and they're feeding because they're drawing fluid can cause wilting, and you can also see yellowing associated with their feeding. They typically can reduce yield, but typically do not reduce quality. An important thing about piercing sucking feeding is that as part of this feeding, they're, we'll say, inserting a straw and then injecting saliva into that straw before they actually begin feeding. When they inject their saliva, they also have the potential to inject pathogens like viruses. Adults are relatively large by aphid standards, and looking at the top left, you can see a winged adult. The top right, you can see that there is a green morph and a pink morph. The green morph is much more common, but there's still the possibility to see the pink morph. Some aphids hunker down on a pod, and the photo bottom right is a female aphid producing a young aphid nymph. So once again, adults are large by aphid standards, 3 to 4 millimeters. Again, highly fecund. They're producing a lot of young, and they can have 15 generations per year. So turnover, population increase can be very high. You're only going to see females in the spring or summer, and this is because they use a life strategy that incorporates parthenogenesis. That is, the females are giving birth to clones. No fertilization of the egg required. So she's giving birth to live young through a process called ovoviviparity, and these young will be produced at a rate of about 4 to 12 per day, and each of these females is capable of producing young of its own in 12 days, 7 to 12 days. So the potential for population increase is great. We'll talk a little bit in the next slide about their life cycle. So beginning on the left side with an overwintering egg, this is going to produce an asexual female. Once again, these are going to reproduce through parthenogenesis or cloning and continue to produce asexual females, as many as 15 generations. And of course, generations, because they're continuously producing, generations can overlap as well. So you're going to have high numbers throughout the season. Winged females can be produced anytime during the course of the year if they experience crowding or if host plant quality decreases. This, of course, is a strategy to move to a new location. Once day length shortens, it influences their endocrine system, so influences their hormonal system. And the strategy to cope for an upcoming winter and harsh conditions is to switch to a sexual mating strategy. So that is, they're going to eliminate the one chromosome and produce males. These are going to produce gametes, that is, sperm cells, mate with females to produce eggs. And the cycle continues. So scouting for Pea Aphid, check at the beginning of flowering. Look at 5 locations per field, and try to keep at least 50 meters between locations. You want to get something that's representative of the field. So, you know, don't pick highlands, lowlands. Try to get something that's pretty ordinary in the field as far as your sampling locations. There are a couple of ways to do sampling. One is to examine plant tips I'll talk about that at the bottom of the slide, and others to do sweeps. And you can do counts based on sweeps, or you can do aphid volumes if you just group together all the sweeps. So you want to look at control based on volume. You look at this table here, and it's measured in cups. It doesn't indicate if that's metric or imperial cups. A cup is a cup, I think. So if you're looking at half cup or higher in 20 sweeps, and so that's a moderate to heavy population, and you got fewer than 10% parasitized. And if you look at the top right photo, you can see these what look like aphid mummies. These are called aphid mummies. Very creative, yes. These are what's left over from parasitoids emerging from mummies that have been attacked. If you see fewer than 10% of those, in addition to a moderate to heavy population and few natural enemies, you may want to consider control. Alternately, you can examine 5 plant tips, so that is the top 20 centimeters, and conduct aphid counts. And the next slide, I'll talk about some specifics associated with the different sweeping versus counting techniques. So, pardon me. So for lentils, consider insecticide application if you got 30 to 40 aphids per sweep, and you have few natural enemies, and the population is sustained or increases over 2 days. That indicates that the natural enemies aren't really having an effect, and you're going to have to inflict some control on this population to keep it from doing damage. For field peas, consider insecticide application if you have 2 to 3 aphids per plant tip, or 9 to 12 aphids per sweep. So that isn't, 9 to 12 aphids in one sweep, voila, we're done. That's on average for all of the sweeps that you've done as part of your sampling. So chemical application when 50% of plants produce some young pods, and there are some assumptions associated with this economic model presented below. The first is that the crop is worth at least $5.71 a bushel. The second is that the cost of control is $6.73 to $9.25 an acre. That isn't just the cost of insecticide, that's all the costs associated with control. So that's labor, fuel, and any other cost that goes into the cost of control has to be factored into that. This economic model also assumes century field peas, and there are other varieties that are more tolerant. For example, pardon me. I'm just going to switch screens that you can't see for my cheat notes. Tipu expresses anti-zoonotic resistance, so that's some repellents, and Trapper seems to be highly tolerant to aphid feeding. So the economic of aphids at 25% flowering is presented in this table. And you can see that it's pretty much a linear scale. So that is more aphids, more economic damage. Not a great leap in logic, I think. But just to give you some specific numbers as far as yield loss associated with specific aphid population densities. I'll just give you a moment to look at this, and then I'll move on to the next slide. Another potentially important pulse pest is the soybean aphid, Aphis glycines, and what's distinctive about these animals is the yellow-green coloration, and you can see the photo on the bottom right. The blue arrow is pointing towards a structure called a cornicle, which is in part responsible for releasing alarm pheromone. These are typically black in these animals. So the yellow-green coloration coupled with that black cornicle is a pretty good field diagnostic for this aphid. This is an invasive insect, was first documented in North America in 2000. No record of it overwintering in Canada to date, so populations that are coming in are blowing in from the States. These can appear as early as late June and are occasionally an economic pest. Once again, highly fecund, make a lot of young, and colonies can double in 1.3 days. So you can get real high turnover, real rapid increase in populations. Much like most insects that feed on fluid with the piercing, sucking mouth parts, plants' juices aren't really nutrient rich. So the insect needs to consume a large amount of this material to get what it needs, and so it's passing a lot of it out the other end. And this fluid that it passes out the other end is a really good substrate for mold. And so in addition to the feeding issues and the potential for vectoring viruses with fluid feeders, you always have the potential for sooty mold growth on plants as well. They are phloem feeders, so that can interfere with photosynthetic pathways. They can also vector viruses, including the soybean mosaic virus. Symptoms include stunted yellow plants, as you saw with Pea Aphid, and leaf distortion in this case as well. Feeding typically can reduce plant growth, pod and seed number, seed weight, and oil concentration, and under high pressures can reduce yields by as much as 40%. So looking at the photo to the bottom right, you can see a wing adult and a nymph, and what you might typically see on the lower leaf in the field, the top picture. Scouting should be soybean crops, should be monitored weekly between R1 and R5. Count aphids on 20 whole plants at 5 locations per field. Make sure these sites are about 20 meters apart. And once again, try to get a representative sampling of the crop. Don't, try to avoid areas that are atypical. You want to get a whole field view of what the insect population is. A W pattern is recommended for sampling. So each of the dots represents a sampling spot. Walk 20 paces, another sampling spot, walk 20 paces. So the action threshold associated with soybean aphid is 250 aphids per plant. This assumes 80% of the plants have aphids and the plants are not yet at full seed. And it also assumes that populations are on the rise. If populations are holding steady or decreasing, this indicates that natural enemies are having an effect. The economic injury level for this insect is 675 aphids per plant. So at this action threshold, this gives you about a week to effect control if the population is still increasing. Natural enemies of aphids. I'm sure many of you are familiar with these. We'll run through them quickly, starting with top left. Lacewing larvae, and here you can see dispatching a caterpillar. A lot of predators are generalists, and as you can see, a lot of predators can dispatch prey items that are considerably larger than they are. Some also have interest in prey items that are smaller, and typically this list includes those. So the lacewing larva, the lacewing adults, very conspicuous, very distinctive. I'm sure many of you have seen these in the field or in your yards or gardens. Lacewing eggs are also highly distinctive, and you can see it on the lower surface of this leaf on stalks. Top middle is a hoverfly adult. Again, these are pretty ubiquitous. You're going to see them in a lot of different regions, and characteristic in part by their flight pattern. So their pattern of hovering, moving rapidly to another location, hovering, moving rapidly to another location. Not many things fly like they do. Their larvae are voracious predators, and here you can see one munching down on an aphid. Damsel bugs are also voracious predators, and here you can see one laying a hurting on a lygus bug. So once again, they can take down prey items that are larger than they are. They're also interested in smaller ones like aphids. Predatory mites are also important predators of aphids, and in this case, this one is munching on a bark louse. You'll see these moving around the field. These are the little red dots you see frantically moving around in what seems like random directions. What they're doing is just casting themselves around until they bump into a prey item, then they attack it. Minute pirate bugs are also important important predators of aphids, and here you can see one munching on an aphid. They take the term minute quite seriously. They're very small. typically less than 2 millimeters long. And the final photo, bottom right, is a ladybug adult. Cute as a bug's ear, unless you're an aphid. Then they're a bit of a horror show. And here you can see one munching on an aphid. Another pulse pest that can be periodically important is two-spotted spider mite, Tetranicus. And the top right photo is an extreme close-up. These are very, very small animals. Some of the damage associated with these beasts, the photo below, and these are the chlorotic mottled leaves. These mites have a very broad host range, in excess of 200 potential host plant species. They are very small, and they can be very numerous, so a number into the tens or hundreds of thousands in individual colonies. Their webbing is characteristic, and you'll see these on indoor plants, greenhouse plants, and field crops. They are distantly related to spiders, so they do produce they produce silk in an analogous manner to spiders. They don't use their webbing to catch prey. Mites are really sensitive to relative humidity, so what they do is they use the webbing to maintain climate control, so that they feed. Mites are susceptible to relative humidity, as I mentioned before. If they lose turgor, they actually absorb water through their cuticle and through specialized structures. If they lose turgor, they actually can't stick their mouth parts out and feed. They're feeding, they rasp with their chelicerae, their mouth parts, and so their feeding destroys cells and can produce this chlorotic mottling of leaves and contribute to leaf senescence. So scouting, look under leaves, and once again, these beasts are very small. Their webbing is very characteristic and very conspicuous. You can take tap samples, that is, place a clipboard or tray below a plant. You can even use your hand in a pinch. Give the plant a tap and try to dislodge the mites. Once again, these are very small, so it's probably a good idea to have a little field lens with you. And then you can get a look at them in their distinctive two spots. There are no published thresholds for field crops, but there are under heavy pressures, which nominal pressures. There are products registered for soybean and alfalfa, and these include malathion, a couple of dimethoate products, oberon, and an abamectin product. You note that there's a couple of asterisks there, and reason being is their natural enemies include predaceous mites, which are also highly sensitive to these insecticides. So if you apply an insecticide, you're really hitting their natural enemy complex very hard, and the harder you hit it, the greater potential for a flare-up of spider mites, because you've really knocked down the natural enemy complex. So the reason for the asterisks is these are a little bit easier on the natural enemy complex. I'm actually reluctant to even call these a pest, but they're an insect of interest and an insect of note, and I've gotten a couple of questions about these this spring. These are agromyzid leaf miners, so that is, they're flies in the family Agromyzidae. You look at the photo on the top right, and you can see that there are the characteristic mines associated with the larvae. I'm seeing, to say a fair number, I'm seeing a decent number on seedling peas this spring. These are attributed to both a fly in the Phytomyza and about 3 flies in the Liriomyza. So it's a complex of different species, all result in similar damage. This is a Liriomyza pictured just below that's the adult fly pictured just below the leaf. The damage associated with these is not thought to be economic. So the loss of older leaves shouldn't impact yields, and typically they're going to be laying their eggs earlier in the spring, and the leaves that are lost probably shouldn't influence the vigor of the plants. Another important consideration is that they are well controlled by eulophid parasitoids. That is wasps in the family Eulophidae, and that's pictured bottom right. Greatly magnified. These are tiny little animals, but important parasitoids of agromyzid leaf miners, and effect great control on their populations. Important to talk about cutworms, and I'm just going to talk about one in particular for no particular reason. There are, about 5 species that can be economically important in a number of different crops, including pulses. So I'll talk about Dingy cutworm, which is a complex of 3 Feltia species with slightly different geographic representation. All are North American natives. All overwinter as larvae, as you can see from the figure at the bottom. These eggs are laid near host plants in the fall. They consume a little bit, overwinter as larvae, complete their development by munching on crop plants in the spring. Once again, very broad host range. They will affect pulses, canola, some cereals, and most damaging in the spring. Larvae are defoliators, and unlike some other cutworm species, rarely cut stems. They will actually climb plants. So the top right is a photo of the larvae or the cutworms, and the photo below that is an adult. And I'm sure most of you have probably seen these flitting around your porch lights at one time. So I'll give you a moment just to take a look at their life history associated with different life stages. So once again, they pupate through the summer, emergence of adults in the late summer, oviposition, and overwintering of larvae. Scouting for dingy cutworm and several cutworm species are generally associated with growing areas of bare soil after crop emergence. If you see these, there's an indication you may have a cutworm issue. There's not much point in sampling right in the middle of a bare patch. The insects have moved on to where the plants are. So concentrate your efforts on the margins of these bare patches and look for larvae there. You're looking at taking a sample of the top 2 to 6 centimeters of soil. No need to dig any deeper, but you do need to dig, so have a shovel or a trowel with you. Look at a 50 by 50 centimeter area, multiply this by 4 to get a per meter count. Repeat this 5 to 10 times every 50 meters, and you'll get a good idea of your whole field population of cutworms and whether or not you need to be thinking about control. Thresholds for peas are 2 to 3 larvae per square meter in the top 10 centimeters of soil. So once again, don't dig too deep. There's no need. The insects don't go that deep. A lot of natural enemies associated with cutworms, and I'll talk to this point brief in just a moment. A lot of products registered for chemical control. Once again, see the crop protection guide for recommendations for that. Once again, lots of products, including BTK bioinsecticide, which would arguably be much easier on the natural enemy complex. Greater expense associated with it. There's always a balance. So beginning with the top, you can see a cutworm larvae with parasitoid larvae emerging from it. The bottom left, you can see an ichneumon parasitoid stinging or parasitizing a caterpillar. So what she is doing is injecting eggs and venom into the caterpillar, and the eggs develop inside the hemocoel of the host insect. So you think of a caterpillar, it's almost like a hollow sausage. Everything's sort of attached to the outside, and it's basically a bag of blood in the middle. And inside this bag of blood is where these larval wasps are developing, taking in nutrients from the fluid. So not actually consuming tissue, but taking in nutrients from the blood. The top right. Once they complete their development, as you see from the top middle picture, they chew their way out. They're going to spin a little cocoon and complete their development there and emerge as adult wasps. The top right is another parasitoid. In this case, it's a fly, a tachinid. What's distinctive about these? If you look at the abdomen, just the large coarse hairs sticking out of the abdomen are quite distinctive. An adult Ichneumon wasp, this is Bancus. This is one that you'll see quite frequently. You'll note it's posed on canola here. You will also see this in pulse crops. Bee flies are also important parasites of cutworms, and that's as larvae. So they will lay their eggs near where cutworms are in the soil, and these larvae will attack the cutworms. Ground beetles again, and you can see the center bottom picture of a bit of a tag team of ground beetles working on a cutworm. So important to consider your natural enemy complex when you're applying insecticides. If your natural enemy complex is large and diverse, you may want to hold your fire for a little while. And there are some other specific methods recommended with the Ag Canada Cutworm book that I can provide a reference to later on. In an effort to get through this presentation in a timely manner, we're going to go on with the next slide. This is an insect that is eliciting a bit of attention recently, and this is less a field problem and more of a storage problem. And this is the bean weevil. And unfortunately, common names being common names, this isn't a weevil at all. It's actually a member of the leaf beetles, the Chrysomelidae. So strictly speaking, it should be called the bean beetle, but there are other bean beetles, just to add to confusion. Medium-sized beetle at 6 to 7 millimeters. What's conspicuous about these guys is if you look at the top right photo and the bottom right photo, is the last abdominal segments are sticking out from the elytra. They will feed on foliage and seeds as adults and larvae in the field, but once again, they're primarily a storage issue. Adults are going to emerge from the spring, feed for a couple of weeks. They can lay eggs on pods and develop within those developing seeds. The larvae burrow into the pods and begin to feed on seeds and emerge after about 40 days. So the top left photo, this is a photo from the government of Western Australia, where this insect is a very big deal and becoming a bigger deal. You can see the hollowed out seed there. So in Canada, it's widespread, US widespread, Africa and Europe widespread, not currently widespread in India. So there are fumigation requirements on the part of the Indian government for import from Canada. So in addition to other trade issues associated with getting your pulses around. We'll talk about the pea leaf weevil survey. This has been ongoing for a couple of weeks now in some areas, still ongoing. This is the map from 2017. So you can see moderate numbers through the south, one real hot spot in the southwest, and very low numbers in the north. So it's as of last year, was still concentrated in the south, working its way eastward. Numbers this year are lower than what we saw last year. Numbers are very low, and we speculated a little bit about this before this presentation began. It's been the subject of some conversation, and we can maybe take up the speculation again at the end of this presentation. So we're going to wrap this up with some recent regulatory developments, and this is associated with the neonic seed treatments. So that is thiamethoxam, clothianide, and imidacloprid. It also has meaning for foliar applications, but what I'm going to talk about strictly has to do with seed treatments. We'll say in the context of pea leaf weevil in peas as an effective control. Most of the products that you're going to come across for seed treatments are going to be thiamethoxam and imidacloprid, and primarily thiamethoxam, to my knowledge, is the one that's used and the most efficacious for pea leaf weevil. So products with these insecticides use the seed treatments for soybean, dry bean, chickpea, faba bean, and field peas. The Pest Management Regulatory Agency has recently issued proposed decision documents related to these compounds, and these are associated with pollinator health and with aquatic invertebrates. And I'll get into the meat of some of these proposed decision documents in just a sec. So for thiamethoxam and clothianidin, pollinator health. The Pest Management Regulatory Agency has determined that water sources do not pose a risk of concern to bees. On a happy note. They also propose determine that a negligible risk to pollinators expected for illicit seed-treated crops, and products containing thiamethoxam and clothianidin are being proposed for continued registration in Canada. So, bit of good news. So, the concern associated with these was due to bees exposed. Part of the concern associated with these compounds are associated with bees exposed to the dust generated while planting treated seeds. Most of these cases were associated with corn planting in eastern Canada. As a part of this, the label statement has been included with these products, and I'll let you read that at your leisure. But the take-home message is: use only a dust-reducing fluency agent. Talc and graphite are not permitted. So what the PMRA is proposing at this time is the addition of best management practices, including a statement similar to this one for cereal and legume. Similarly, for imidacloprid, PMRA found that water sources didn't pose a risk of concern to bees, as did seed treatments. The big issues with the neonics seem to be associated with foliar treatments, and that's an issue outside of this discussion, to my mind. Once again, bees exposed to dust are considered an issue outside of foliar application. So there are already label statements in place associated with dust from corn and soybean, and the PMRA is proposing additional label statements for cereals and legumes treated with these active ingredients. So the other issue, so finally, yes, the PMRA's final decision with regard to neonics and pollinator protection is expected December 2018. And once again, the pollinator risk assessment for imidacloprid, clothianidin, and thiamethoxam with seed treatments is favorable for now. So PMRA also issued a proposed decision associated with imidacloprid that proposed the phased out of all agricultural and the major of outdoor uses for imidacloprid over 3 to 5 years, based in part on its toxicity to aquatic insects. So, as part of this, of course, clothianidin and thiamethoxam toxicity to aquatic insects is also a focus. So because of this, a number of different stakeholders are conducting water sampling in order to provide PMRA with good, rigorous data so that they can make a science-based decision based on the data. So proposed decision for clothianidin and thiamethoxam are expected July this year, and that's for the effects of aquatic invertebrates. And a final decision expected by 2020. Imidacloprid still up in the air. And I'm afraid I have to leave it at that. Thank you for listening, and we have a bit of time for questions, I believe.
Fantastic. Just as a reminder, an email will be sent out to you for your CCA number or CCSC number in order to get your credits. And at this time, please feel free to provide suggestions for future webinar topics. We will be in the planning stages for next year's webinar topics and certainly welcome any feedback. A big thank you to you, James, for speaking to us today on insects in pulse crops. And a big thank you as well to Andrea for organizing the session and all of the participants for joining us today. We do have a few questions that have come in, and I would like to remind those of you that are on the line to type in any questions that you may have, and we will address them as they come in. First question maybe for you, James, is you mentioned that there's some speculation as to why we've potentially observed lower pea leaf weevil populations in Saskatchewan this spring, even though we are hearing perhaps of higher populations still in Alberta. Would you like to provide some of your thoughts on why that's happening?
Yeah, it's a really tricky question. There could be a number of factors involved. I believe that there is some talk about the potential effect of dry conditions, both in the fall last year and the spring this year having an influence on populations. To my mind, there's also a potential effect of the hard winter that we had. The funny thing about pea leaf weevil is it's a little less cold hardy than some other temperate insects. A lot of temperate insects and other arthropods can freeze solid and survive temperatures of -18, -20. So the big issue for them isn't really freezing solid. It's more about crystal formation. And so a lot of these animals will produce proteins that reduce crystal formation, or they'll purge their guts to reduce sites of nucleation. Just lots of strategies to reduce the formation of crystals and the damage that they do to tissues. For the pea leaf weevil, the supercooling temperature, so the temperature where crystals form for that guy, is about -7 Celsius. So a little bit less cold hardy than some of the other insects that are either from North America or have naturalized here as pests. So long and the short of it, I don't know what the answer is, but it's pretty good fodder for speculation.
Okay, thank you. Another question, you had mentioned the soybean aphid, that they don't tend to overwinter and that they blow in. Do you have any comments on pea aphid and if they overwinter here or blow in, and when should we be watching for those?
My understanding is that there is some overwintering here, but they will also blow in. I think best to start watching for them probably early flowering.
Okay. You had mentioned pea leaf weevil feeding on the nodules on field pea. We have some observations on faba bean as well. Do you have any comments on if they have a preference for faba beans or peas, or differences between those crops?
Yeah, the feeding preference, for the adults, they do seem to like field pea and faba as a food crop for the adults. But as far as larval development, field pea is a far superior host. So they do much better as larvae on field pea.
Okay. We have seen high numbers of Pea aphid and faba bean. Again, we had calls on that last year. Do you think that there is any yield loss associated with heavy Pea aphid infestations in faba bean at the early potting stages, or any thoughts on potential thresholds in faba beans?
I'm afraid I don't. It seems reasonable that obviously that high populations could be influencing yields, but I don't have any numbers to refer to.
Okay, and another question was on the nominal threshold with aphids in lentils, so pea aphids in lentils. Do you know or have any feeling on the potential yield losses in lentils at sort of that 30 to 40 aphids per sweep threshold?
I'm afraid I don't have any hard numbers for you.
Okay. Do you know, like, if there's just been sort of feedback on potential yield losses observed in lentils with the aphids? Have you seen any of that or heard from people that they have?
Yeah, what I've heard is yes. Unfortunately, I don't have any hard numbers for you.
Okay. I think that is all of the questions that we have had come in. So, James, once again, I would like to thank you very much for your time and effort in presenting to us and everyone on the line today. And again, a big thank you to everyone for taking time out of your day to join us and learn more about scouting insects and pulse crops. Again, this will be posted to our YouTube channel if you have any interest in coming back and listening to this again, or going back and seeing some of those charts and numbers that James referred to today. So thank you all very much, and enjoy the rest of your day.
Thanks all.